Okay. Yes. And so now we're ready to proceed. The next talk comes from Mary at The Ohio State University. And Mary is a postdoctoral researcher in the Injury Biomechanics Research Center at Ohio State. And she's been using Dragonfly back since 2018, so she's got some longevity in her Dragonfly expertise. And when I asked how she learned about it, she found it in the scientific literature from other people. So please keep publishing your work, and when you tell people about Dragonfly, you'll spread the word. So let's give Mary our attention while she tells us about three-dimensional visualization of bone, tissue, vascular, and cellular spaces.
All right, thank you very much. I'm going to talk today about some of the ways that I use Dragonfly primarily in visualization. I'm definitely interested into getting more into segmentation, but traditionally all of our work has been done in ImageJ and Fiji, and also in some CT analyzer products. So a lot of my work started out doing the segmentation there, but I ran into some problems I couldn't solve there, so I've started moving over to Dragonfly. And my vision is to eventually do everything completely in Dragonfly and have it be essentially completely open source and accessible in sort of a package. But today I'll talk about some different ways we can visualize spaces within bone tissue and what that can tell us about bone health. So health is something that I study in the context of human variation. My background is in biological anthropology. And basically what that means is that I look at human variation as not only biology, but also the intersection with environment and behavior. So your body is not only your biology, your genetics, but also what you encounter over the lifespan and how you behave over the lifespan. In addition, my focus is on skeletal biology, so I look at sort of a finer grained bone structure as a combination of physiology and mechanical adaptation, which are two of the main things that drive bone structure adaptation over the lifespan. And specifically at the level not of the sort of gross morphometry of the bone or the functional anatomy of the bone, but at the bone tissue level. So looking at not only the cross-sectional geometry or the traditional sort of trabecular architecture, but looking specifically at vascular pore networks within cortical bone. And this is relevant for understanding bone loss and bone fragility, targeting vulnerable populations who, because of their physiology, such as age or pathology, may be more vulnerable to bone loss or to bone fragility, and also to looking at-risk tissue sites and the timing of intervention so we can hopefully eventually treat bone loss before it occurs rather than sort of when it's irreversible. A little bit about what I'm going to go over in this presentation. I'm going to talk about different ways we can look at bone quality through different aspects of bone tissue. I'll talk a little bit about 3D imaging methods, which will probably be familiar to most of you, micro-CT, SRU-CT, and confocal laser scanning microscopy, which are the main ones that I've used for visualization. And finally, I'll talk about two projects that I've used for visualizing these spaces, looking at mechanical strain patterning and looking at age-associated changes in these spaces. So first of all, what do we mean by bone quality? Bone is affected primarily by mechanical adaptation, but it has physiological modification of how well it can sense and respond to those mechanical drivers. And the cells that are embedded in your bone tissue that sense this mechanical demand, both strain changes and damage, are osteocytes, which you can see here. And they trigger the removal of bone through osteoclasts or the formation of bone through osteoblasts, which we've already heard a little bit about in presentations today. And there can be two ways that this occurs. There can be a separate removal or formation of bone called modeling. So if you think about just your ordinary bone, it encounters high strain. It will have apposition externally until it returns to deforming at its customary level. So if the bone has sort of a set point of how it would essentially like to deform such that its osteocytes are no longer being signaled to add more bone, it will add bone until it's no longer out of that, in that range. So you can see here, this is a cross-section of a rabbit femur that has been stained with calcium during remodeling activity, and you can see those rings are modeled apposition. And it primarily occurs externally on the periosteum, but there can also be drift or movement of the bone, so you also see it internally at the marrow cavity. Modeling can also sort of counterintuitively involve resorption, at least in some of the ways it's been conceptualized. So where there is low strain, osteoclasts will be signaled to remove bone, primarily internally at the marrow cavity, but again, drift can occur. However, what I tend to focus on is the activity that occurs within the cortical bone itself, which is called remodeling. So you already have bone there, it's either old or it is damaged, and so the osteocytes have signaled for that bone to be dug out and replaced with new bone. And this occurs through the formation of a tunnel called a cutting cone, where osteoclasts tunnel in and remove the bone, and osteoblasts come behind and fill it in, and it has to transmit a blood vessel because otherwise the osteocytes that become embedded there will die. So they need the blood vessel to have that pore to come through and nourish them, and also to have that as a branching off point for additional remodeling. And so this can occur in sort of two ways. You can have it triggered by damage to the bone, which is called targeted remodeling, and that would occur under high strain. And you can also have it occur during what used to be called stochastic remodeling. I think there's a movement towards calling it disuse mode remodeling because there's always a little bit of bone loss. So it's not truly stochastic, but under low strain, the bone will either turn over or even experience some minor bone loss. And again, this is so that the bone returns to its customary deformation. And you can see here the calcine labels on new bone that was formed over this period within the rabbit femur. So this is not something that occurs consistently over the lifespan, and in fact it is modified by aging and other types of physiological changes. Age-associated bone loss begins a lot earlier than people think. Bone resorption begins to outpace bone formation as early as your 20s for trabecular bone and your 30s for cortical bone. So bad news, it's already going, and in some cases gone. So what can we do to sort of detect and quantify this bone loss? Well, this is a problem that is, I like to call, it is the universal human pathology. In the United States, as we've already heard, osteoporosis is very pervasive. Adults over the age of 50 are clinically diagnosed with low bone mass or osteopenia, and high bone mass or low bone mass with risk of fracture or osteoporosis at strikingly high rates, slightly higher for women. And this higher incidence in females is due to a perimenopausal loss of estrogen, which is a sex steroid that preserves bone. So if we look at the femoral neck, here are some changes in females and in males over the lifespan. Two of my pictures did not show up. And so, as we've already heard, the clinical evaluation for this is a DEXA scan, but this is under predictive of fracture risk. So this is something that, again, compares your two-dimensional, essentially, aerial bone mineral density to a standard for a population and sees if you're unusually low. But ABMD declines between age 60 and 80 only account for 2 times of the overall 13 times increase in fracture risk. And moreover, if you look at hip fracture patients, they often don't even reach the BMD standard for osteoporosis or osteopenia. So about 54% of people who actually have a hip fracture are not even diagnosed as osteoporotic. So clearly there's something more going on than just bone mineral density. And so what we can look at is not only sort of the material composition of the bone, but also the microstructure. And bone tissue microstructure collaborates in a hierarchical system to support bone strength. So again, looking at that femoral neck and taking a section off, there is a collaboration sort of down the hierarchical scale between the cross-sectional geometry, trabecular bone, the porosity, the osteons that surround the pores, osteocyte lacunae, and all the way down to canaliculi and microcracks at almost a nanometer scale. So different ways we might quantify bone tissue quality: the cross-sectional geometry, the area and the distribution of the shell of bone. Again, our cutting cone picture, cortical pores, which are required for nutrient transport, and these can be seen in both 2D and 3D. Secondary osteons, which surround the cortical pores, here in a rabbit femur, highlighted with white field fluorescence. Osteocyte lacunar canalicular networks, so the mechanosensory bone cells exist within their own pore networks, and changes in the density and communication of those can affect how well the bone senses and responds to strain. And finally, microdamage, microscopic cracks, which allow us to diffuse damage and then repair it without fully fracturing the bone. This is something that humans are particularly good at. And you can see this with a variety of different 3D imaging methods. And then finally, the traditional trabecular architecture changes in the cancellous bone within the marrow cavity, which has long been the focus of 3D imaging just because you can see it at a lower resolution. And so we're only really starting to push into looking at some of the structures in that cortical bone tissue as well. And here's an example of changes in trabecular architecture. So changes in bone quality with age increase fragility. You have a decrease in cortical thickness, the trabecular architecture, and the osteocyte lacunar canalicular network. And concurrently, you have increases in cortical porosity, microdamage, and collagen and mineral brittleness. And so these things are sort of occurring simultaneously, where you have less bone and the bone is also worse. So how can we actually visualize some of these changes? The traditional thing you might get, if you had an injury, it would be a CT scan, and we can actually dial that down and do a micro CT scan as well at a much higher resolution. And there is always this trade-off between, as we just heard, field of view and resolution. If you want to see something at really high resolution, you're going to have pretty much a small field of view, right? So the closer we get to the very tiny structures, the less of those structures we can see overall. So here's a comparison of an HRPQ CT. This is actually a proxy, but it's the same resolution from a CT scan compared to micro CT of the femoral neck at variable resolutions. The highest resolution imaging method that I've used that is also an X-ray based method is synchrotron radiation micro CT, and this accelerates electrons close to the speed of light, allowing us to achieve submicron resolution, and this has to be performed at a specialized light source facility, which is essentially just a giant microscope. And I did this at the Canadian Light Source with a postdoc collaborator, the Andronowski Lab, which I'll talk about a bit. And this can see much higher resolution structures, not only those pores again, but also the osteocyte lacunae where the cells live, as well as microcracks and with phase contrast, even secondary osteons. A way that you can get at these structures that are around the cells of bone in your lab is with confocal laser scanning microscopy. And this essentially is a type of fluorescence, wide field fluorescence, that blocks out of focus light either with a pinhole or a spinning disc. There are other ways of precisely exciting only a very small amount of the fluorescent stain with multiphoton confocal microscopy. And this allows you to get very precise resolution in X, Y, and Z planes, and then you can stack those Z planes into a 3D image. But it can't get very deep in bone. The deepest I've gotten is about 100 microns. So again, that's your trade-off. So I'm going to talk a little bit about two projects that I've used Dragonfly for in various capacities for visualization and some processing. This is from my dissertation work, and this is something that I found Dragonfly towards the end of when I was stuck on a problem. So it is femoral neck segmentation, but it is through morphological smoothing and binary operations. So my goal is to move towards the deep learning and machine learning, but you'll see it's a bit more brute force than what we've heard so far. So why do we care about mechanical loading history? Microstructure is essentially a record of how the tissue was loaded and how it responded to that loading. Bone adapts to mechanical demand over the lifespan, and we can then look at how aging and pathology dysregulate this adaptation. So a key example, again, cortical porosity. Having a hole in your bone is actually necessary if you want to be able to fix your bone, because if you don't have that pore, then the blood vessel cannot be transporting the nutrients in the cells. So porosity is sort of a necessary evil, and like microdamage, in normal quantities, it's not actually damaging to the bone. However, as you have a dysregulation with age, where you're resorbing a lot more bone than you form, and the cells aren't as good at sensing how much bone needs to be formed, you get this accumulation of vascular porosity. And these holes become stress concentrators. And this means that those microscopic cracks that we use to diffuse energy in the bone can actually get into those pore systems or initiate in the pore systems and propagate into an actual fracture under high strain. So for example, an increase of only 4% porosity leads to 84% increase in crack propagation. And 76% of age-associated loss of strength in the femur is due to cortical porosity. However, again, there are some complications in inferring mechanical loading history from remodeling. First, As we already heard, remodeling is triggered by both low strain and high strain. So how are we going to tell what kind of strain it was under if the same process can essentially result from opposite ends of the spectrum? How do we distinguish between targeted remodeling and disuse mode remodeling? And this is exhibited by the fact that several studies have found no significant intraskeletal differences in percent porosity between bones that experience highly different loading sort of mechanisms and amounts of load. So the rib, the femur, the tibia, and the distal radius, histologically, have all been found to have essentially equivalent percent porosity. So obviously there's some sort of other regulation going on other than absolute strain. The other complication is that physiological dysregulation by aging is highly individual specific. Age, sex, and BMI only account for 12.1% of 2D porosity in the midshaft femur. So instead of asking, can we predict the absolute amount of porosity from age and some other factors, let's ask, can we distinguish what type of remodeling is actually responsible for this porosity? In other words, are pores produced by high-strain targeted remodeling morphologically different from pores produced by lower-strain disuse mode remodeling? And does this alter with age? So to look at this, I looked at an age-matched series of 7 male and 7 female postmortem human subjects, comparing the right femoral neck, which is weight-bearing and dynamically loaded and commonly fractured, to the rib, the right fourth rib midshaft, which is comparatively unloaded. I chose the femoral neck because in humans it has a predictable strain gradient where compression is almost zero at the superior and then increases towards the inferior. So this would allow me to look at how pore systems were changing morphologically from superior to inferior as strain increased. The rib is a little more complicated because there are different ways it can be loaded depending on its sort of level in the thorax. If you divide it into the skin side, the cutaneous, and the lung side, the pleural, and you have a lateral expansion of the ribs from an inhalation of your lower ribs, you place one side in tension and the other in compression. And this is supported by a higher percent porosity in the region that we think may be under tension. However, if you go higher, the inhalation actually switches the way these ribs are moving and should place the skin side in compression and the lung side in tension. And then finally, if you have a forced inspiration, these mechanisms can occur simultaneously. So the rib is a little more complicated in modeling its mechanical strain patterning. So I wanted to sort of use the femoral neck and its patterning to see how pore systems should look under variable strain, and then sort of use that as an atlas for what the rib is actually doing. So this is where Dragonfly saved my life, essentially. Because of the high cost of micro CT scanning and the fact that I was a PI on my own grant and I had limited funds as a grad student, I could not do the sort of scan the entire femoral neck and then zoom in on ROIs. It was too expensive for me. So I had to physically cut it into 3 pieces. And because I was using a helical based micro CT scanner, the sample could be as tall as I wanted it to be as long as it fit within the field of view, which was about a 10 by 10 millimeter cube. And so I had to cut each femoral neck into 3 pieces and stack them on top of each other, and here they're wrapped in a parafilm and saline soaked gauze to keep them moist for BMD. And then I was able to scan them at 6.4097 microns. And automation was really essential here because each image stack was 1,561 transverse cross-sections. So even though I didn't use deep learning, I had to do some sort of automation for consistency and also so I wasn't hand outlining things. So I developed, again, prior to discovering Dragonfly, I developed sort of 2 helper tools to do this. I developed a macro set for ImageJ or Fiji called Pore Extractor 3D, and then I developed a number of routines for CT Analyzer, which is what our lab uses for analyzing morphometry in our CT scans. So I wrote first a Fiji macro that picks essentially the best grayscale pixel range, which changes over the scan, the 20-odd hour scan of a column of a femoral neck. You can't use just one grayscale value, you have to write an algorithm for it. So I did that. This is where Dragonfly saved my life. Because I had to physically cut them into 3 pieces, I then needed to stick the pieces of the femoral neck back together. And so I couldn't just do this with a 3D model because I also needed to look at the grayscale values. Because if you orient something after you've binarized it, it's no longer binary because it's creating all of these intermediate grayscale values. So I had to be able to load it in the grayscale which was about 60 gigs of data, and orient it so that I could then binarize those individual pieces and stick them back together. Because my computer wasn't good enough to adhere them, and they also didn't overlap at all. So I basically had to sort of align them and then put them back together. So Dragonfly allowed me to visualize this both in 3D and then in every sort of axis to ensure that the stacks were where they were supposed to be aligned. I then conducted adaptive thresholding to be able to isolate the small pore structures and trabecular margins without losing them. And I'll just go through sort of some of the steps of that and remove the mounting fixture with the mask. I also conducted, I created an algorithm, or a macro rather. Let's see if this will play. Maybe not. To essentially, after I had those binarized slices, I created a little macro in Fiji to look at the 5 sort of slices throughout this long stack and allow you to manually stick them back together, essentially, by looking at 5 waypoints along that long stack. So that's what this is doing. It allows you to position them all simultaneously, make sure they fit, and then stick them back together. So obviously it would be preferable to have an overlapping scan that could actually be merged with computer applications rather than manually, but this is sort of what I was stuck with, and so I worked with it. And then finally, the way that I isolated the cortical bone from the trabecular bone was essentially by extracting the marrow cavity and smoothing it out a lot. And again, this isn't going to be as precise as deep learning because you run into that issue where you have holes very close to that cortical trabecular margin that look like the holes that are inside the cortical bone. And so that differentiation is a little less precise, but it does work pretty well for ribs and fairly well for femoral necks as well. And essentially this is just extracting the marrow cavity and smoothing it out consistently. And then you can use that endosteum as a mask to extract a number of different aspects of that binarized bone. I also differentiated between cortical and trabecular pores based on distance from the marrow cavity. And I'm just going to skip these videos real quick. I created regional rib masks as well and femoral neck masks to allow me to look at the quadrants of the femoral neck and the halves of the bone. And then finally conducted 3D morphometry and CT analyzer. So here's an example of a young female compared to an old female. Sorry. And a young male compared to an old male. And something that you might notice in the female is a very strong apposition of bone tissue, which I'll talk about in a second. But what I found when I sort of mapped all of the variables is that they were all following a gradient of either increasing or decreasing inferiorly under high strain. So all of the graphs of the means of these variables essentially look like the parabola that is the bottom of the femoral neck. And so the femoral neck region statistically confirmed this structure strain model where under lower strain there is disuse mode remodeling resulting in large interconnected obliquely oriented pores. Whereas under high strain, very targeted, brief, precise remodeling, you get small, isolated, longitudinally oriented pores. The remodeling isn't as permissive, it gets in, it gets out, and it makes these very tight, isolated pore systems. And this was dysregulated with age by increases in pore density and decreases in pore separation, implying pore convergence. Pore thickness was often localized to the superior anterior octant, which has also been found in previous studies. And this is, unluckily, the region that is particularly vulnerable to crushing during a sideways fall. So vulnerable individuals who have this apposition in old age may be particularly vulnerable to fragility fractures from just falling over sideways because there's a giant gap right where that compressive stress is at its maximum. The rib regions also matched this high strain morphotype in the pleural cortex with the traditional model where the lung side is compressed and the skin side is tensed. But it was a little more dysregulated. There was less consistent regional patterning in some of the variables, and it was more sensitive to age and cortical thickness. Just briefly, we also examined this in opioid-driven bone loss using a rabbit model, and used the same sort of macro set to visualize this. and found that opioid use tended to make the bone insensitive to differences in mechanical loading between the femur and the tibia, resulting in bones that had very similar morphometry rather than being adapted for their distinct purpose within the rabbit's locomotion. All right, I think I might be out of time. Okay. Let me just show you this real quick. This is some visualization I did with synchrotron radiation micro-CT using a very similar macro set in addition to some aspects to correct for the, sorry, one second. Oh, it doesn't want to load them.
It's a low power laptop.
Yeah.
Struggling to keep up.
Okay, I'll just go past that. I think I might just end it there and take any questions that you might have. Sorry, the videos. Seems to be switched to a different presentation.
Oh, I think PowerPoint gave up.
Oh, okay.
All right. Well, thank you very much, Mary. That was very exciting, and it looked like another heroic data analysis effort. And I want to say that it was. I would love to take credit for choosing and organizing the speakers in a coherent way, but it was purely serendipitous that we get to see back to back how poor systems tell us about loading history and age-related pathology, and how poor systems tell us about how hot your fire was when you baked your pottery 1,000 years ago. Having said that, do we have time for maybe one or two questions for Mary, and then you'll be here over the lunch hour, so you can catch her at lunch. Do we have any questions from the audience you want to get in at this time? We do. Let's go back over here. Yeah.
How did you measure the strain?
How did you measure the strain that you're reporting?
That's a great question. So this is purely.
This is the Zoom microphone.
This is purely just from sort of looking at the anatomical configuration of the bone. That's why I picked the femoral neck specifically, because it has more predictable loading. Many times people will use like the midshaft of a long bone that experiences variable loading modes and variable loading magnitudes throughout a walk cycle. So I like the femoral neck in humans because it's sort of more consistently strained. Our upcoming project is we're going to be working on scanning a lot of bones that have actually been mechanically tested with strain gauges so we can measure the strain and see if it actually corresponds to what we're seeing with porosity.
All right, I think we're going to save the other questions for lunch. Let's thank Mary again.